The main advantage of this simple technique is to generate cardiac progenitors and cardiomyocytes of defined hard field origin and subtypes. In turn, resulting cells can be used for a wide range of research applications, including basic developmental biology, proof of concept cell-based therapy, and cardiac disease modeling studies. Demonstrating the procedure will be Sean Spiering and Michael Yu from my laboratory.
To begin, coat enough wells of a 96 well plate with 50 microliters of coating reagent to accommodate subsequent cell seeding and viral infection steps. Then add 1 milliliter of dissociation reagent to a well in the 6 well plate to detach the pluripotent stem cells seeded. Then, use a sterile pipette to transfer the cell suspension in a 15 milliliter plastic centrifuge tube. In the tube, add 5 milliliters of PBS containing calcium and magnesium to neutralize the dissociation reagent. Then centrifuge the cells at 200 times G for 3 minutes. Post-centrifugation, discard the supernatant, and then gently flick the tube for the pellet to be dislodged. Then dissolve the cell pellet in 1 milliliter of stem cell medium. Next, use an automated cell counter to count the number of cells. Once the counting is done, seed 20,000 viable cells in each well of a 96 well plate containing 50 microliters of stem cell medium supplemented with 2 micromolar Rho-ROCK pathway inhibitor. After 20